My pleasure to introduce our next speaker, Dr. Charlie Gersbach from Duke University. He'll be talking about epigenome editing technologies for cell programming. Charlie.
Okay, thank you very much for the support and the opportunity to tell you about some of our work. We just heard two really amazing presentations about how to image and map the epigenome, and our work in this area is really focused more on developing technologies and applications for making targeted perturbations to the epigenome. And our work in this area has largely been motivated by this famous Waddington landscape. And so the idea, is this making funny noise? So the idea in this landscape is that you have this marble at the top of the hill, and this represents a pluripotent or totipotent cell, and as that cell starts making decisions that are gated by gene expression changes, it starts rolling down this landscape and falls into different valleys in between these hills. And each of these valleys represent a stable cell state, such as a differentiation state or a decision that the cell made. And then, therefore, the hills in between the valleys represent the barriers that prevent one cell from spontaneously becoming another cell type. And so, for example, neurons in our brain don't spontaneously become cardiomyocytes or fibroblasts, largely dictated by these epigenetic barriers. And I think that historically, it had been thought of these epigenetic barriers being permanent, but we know now from the work of Yamanaka and Gurdon and others in the reprogramming field that in fact you can overcome these epigenetic barriers to change between cell states. However, most of the methods that have been used to do that type of cell programming are not particularly specific. These are things like small molecules or growth factors or cytokines that have many different effects across the genome, or overexpression of master transcription factors like OCT4 or others that bind to tens of thousands of sites across the genome when overexpressed. And so what we were really motivated to do was to develop tools that would allow us to ask the question, what are the minimum and sufficient the necessary and sufficient epigenetic changes at specific nodes in the genome that will dictate cell fate decision making. And so the tools that we've been using most recently are based on the CRISPR-Cas9 system. Now, most people are familiar with CRISPR-Cas9 as a genome editing tool. And so for that reason, you see the Cas9 nuclease here is the blue blob, and it makes the complex with the guide RNA, and then goes and finds its target site in the genome where these scissors act as the nuclease that cuts the DNA. Now, we are more interested in the inactivated form of Cas9, or what we call deactivated Cas9, or dCas9 for short. And in this case, we make 2-point mutations to Cas9 that inactivate its cutting activity. It still binds the guide RNA. It still goes and finds its target site in the genome, but the scissors are now broken by those 2-point mutations, and it can no longer cut the DNA. So now this serves as a RNA protein molecular scaffold that you can target to any site in the genome. And then by attaching or recruiting any type of epigenetic modulator or regulator of gene expression or any other molecule, you can localize that molecule to a very specific site in the genome. And in this case, we're interested in making specific epigenetic perturbations, so things like modifications to histones or DNA methylation. Now, we've been working in this area for a while and developed a number of different epigenome editing tools and then used them for different applications. And last year at this meeting, I talked about our work in targeting known factors in the genome that are involved in things like neuronal cell fate commitment, and targeting those to convert fibroblasts into a neuronal-like cell. And so I talked about this last year, but this focused on taking known factors. And in this case, there were 3 different transcription factors that had been previously described by Marius Vernig that we knew if we went and epigenetically activated them from the genome that we might get neuronal cell fate commitment. And we showed that previously, but these neurons were really generated from known factors and gave us things that looked like neurons, but were really immature neuronal cells with immature electrophysiological properties. And the conversion efficiency was not particularly high either. We see here about 4% of these fibroblasts were converted into neurons in response to epigenome editing with our CRISPR tools. So most recently, I want to give an update to this story. Rather than being restricted to the few factors that we know might be involved in directing a cell fate decision, we wanted to develop an approach where we could take an unbiased strategy to scanning the genome for factors and epigenetic nodes that we can target to change cell fate specification. And so the way that we did this original experiment was we wanted to study or to screen all human transcription factor genes that might be involved in human neuronal cell fate specification. And we did that starting with human-induced pluripotent stem cells. And we engineered that stem cell line first so that it had a 2A mCherry reporter downstream of this TUBB3 gene, which is a marker, kind of a pan-neuronal marker. So now in this pluripotent cell, any time that it differentiates into a neuronal-like state, it should turn red. The other thing that we did with this cell line is we engineered it to express this dCas9-based epigenetic activator. But there's no guide RNA, so this activator shouldn't be doing anything in these cells. But we can then introduce a library of guide RNAs with lentiviral vectors such that every cell gets a single guide RNA, goes to a different site in the genome, and recruits that dCas9-VP64 activator to activate gene expression wherever that guide RNA is targeted. We do that so that every cell only gets a single guide RNA, and then after several days, we can sort the cells based on whether they turn red or not, isolate the red cells or the non-red cells, sequence the guide RNAs that are present, and use that to identify factors in the genome that are responsible for pro-neuronal differentiation of pluripotent cells, or, as I'll talk about later, actually repressing that differentiation process. So the guide RNA library that we used was targeted to 1,500 transcription factor genes in the genome. So these are all genes that we found that have been annotated to have potentially some transcription factor function. We had 5 guide RNAs targeted to every one of these 1,500 promoters, and combined with 100 negative controls, that was about 8,500 different guide RNAs that we introduced into that cell line that I described. And then after introducing the guide RNAs, we waited 5 days and then sorted off the 5% of cells that were most red or the 5% of cells that were least red. We can then sequence the guide RNAs that are present in both of those pools and also sequence the starting population and compare them. And what we find is a number of statistically significant enriched guide RNAs present in the most red cells, as well as a handful of other guide RNAs that are present in the least red cells. And so we can look at the identity of those and then go back and test them individually based on what promoter that they target. And one of the nice results was that almost all of these genes reproduce from the screen. So if we go back and target a number of these different genes, we find that almost all of them will activate that mCherry reporter like we expected. But of course, that's only telling us that these guide RNAs are able to make these cells red or turn on the reporter. There might be other ways that the reporter is being activated that aren't related to true neuronal differentiation. And so we take those same guide RNAs targeting the same set of genes and look for actual neuronal gene expression, and we see many of them also activate neuronal genes in addition to the reporter, although some of them do not, and those might be false positives that somehow stabilize the reporter or otherwise target mCherry. I wanted to mention that many of these genes that we found out of these 1,500 transcription factors, we found about 24 that were very robust in inducing neuronal differentiation. Some of these were obvious, things that had already been described, like neurogenin 3. Others were less obvious that we had not been described before as pro-neuronal factors. But in addition to looking at induction of neuronal gene networks, we also looked at morphology and protein staining of different markers to confirm that many of these do induce a neuronal-like phenotype, although some of them, the morphologies aren't necessarily characteristic of neurons, but might be involved in otherwise pushing these cells out of the pluripotent state. So we had this number of different transcription factors that we found that were pro-neuronal, about 26 of them. But we wanted to better understand how they were different from each other and do so rather than just staining for a couple of markers, but looking transcriptome-wide. And so for that, we picked a select few of the interesting factors and some of the well-known factors and used RNA-seq to kind of compare their gene expression profiles. So this is looking at, once we activate these genes in the genome, how does the expression of every other gene across the genome change? And so here we took 2 factors, one neurogenin 3, maybe the most well-described pro-neuronal transcription factor in pluripotent stem cells, and then another factor that came out of our screen, ATO1. And when we perform RNA-seq, we see that there's about 3,000 or 4,000 genes that are changed between pluripotent cells and the cells in the differentiated state. And many of those are shared, but some of those are unique to either one of the two factors. And so we wanted to better understand that. And you can see that across the shared genes, many of these fall into categories that are representative of neuronal differentiation. And there's very good reproducibility across replicates. But to specifically, we then specifically wanted to characterize the gene sets that were specific to either factor. And when we plot all of the gene expression changes when activating ATO1 versus when activating neurogenin 3, we see many of them are shared, but some of them are different. And what's interesting is those different genes fall into specific categories. So for example, the ATO1 activated genes seem to be more representative of dopaminergic markers, whereas the neurogenin 3 activated genes tend to be more representative of glutamatergic neurons. So this got us excited that this type of approach could not only be used to discover new proneuronal factors or factors that could differentiate into any cell state, depending on which reporter you use, but could also be used to overcome some challenges in the field in getting robust differentiation into specific neuronal subtypes. Now, of course, in this case, we just used a pan-neuronal marker for the selection, but we can imagine going back and redoing this for reporters that are specific to the neuronal subtype that you're most interested in. Now, this was a screen that was all based on single transcription factors. I told you every cell got a single guide RNA that turned on one transcription factor in the genome. But we know that most of these reprogramming and differentiation states, they don't result from the action of a single transcription factor. They result from interactions between transcription factors. And so it's very difficult to look at all combinations of all transcription factors. But as a starting point, we wanted to say, OK, let's take some of the proneuronal factors that came out of our first screen that are very robust, and then combine them with activation of every other possible transcription factor in the genome. And so what this looks like is using that same reporter line that I described previously, but now the lentivirus carries two guide RNAs. Every lentiviral particle carries two guide RNAs. One of those is fixed, and we did this 2 times. One where the first guide RNA targeted ASCL1, a known proneuronal factor. Another experiment, the fixed guide RNA was targeting neurogenin 3, another known proneuronal factor. And then the second position in the lentiviral vector was the guide RNA library, targeting all different 1,500 genes. So now in every cell, you're going to get activation of ASCL1 or any other human transcription factor, or activation of neurogenin 3 and any other possible human transcription factor. And then we do the same sorting and sequencing. And now we actually get a whole different set of hits, some partially overlapping, but many new hits came up that seemed to only function in combination with the proneuronal factor. So if we're looking at fold change and abundance of the guide RNA versus significance on the y-axis, these over here would be the pro-neuronal factors. Here I'm showing the results with the neurogenin 3 plus transcription factor library screen. So these are factors that helped make neurogenin 3 even more potent in inducing neuronal differentiation. Over on the left, these were things that would be depleted. So these were things that were antagonizing neurogenin 3 induced neuronal differentiation. And so we wanted to better understand kind of what the hits were that were coming up in this combinatorial screen. We can compare what came up in combination with ASCL1 to what came up in combination with neurogenin 3. Again, you see things that are kind of partially overlapping, but some things that are specific to either one of those two factors. And so we have more work to do to kind of follow up and understand what's going on there. But we then took some of the interesting hits from this combinatorial screen and then again did RNA-seq to better understand what they were doing. And so here I'm showing RNA-seq data for neurogenin 3 alone, or neurogenin 3 with one of each of these 3 different factors that we combined it with. You can see that, again, it's largely overlapping. 2,500 of the changed genes were common across all 4 of these treatments. However, there were many other things that at a particular arbitrary threshold were different between these treatments. And one of the ones that stood out as interesting was this combination of neurogenin 3 with LHX8, particularly because the genes that were specific to NGN3 plus LHX8 were genes that were involved in synapse development. And we looked at that a little bit more carefully and again, saw a number of different synaptic genes that are enriched in the, when we add LHX8 to the NGN3 treatment. And so that's exciting because this might be a mechanism by which we can accelerate the maturation of this neuronal differentiation process, which is a known challenge in this field where people want to use neuronal differentiation of iPS cells for drug screening and disease modeling, but some of these protocols take weeks or months in order to get full maturation of these cells, and that's a real bottleneck in pharmacological development, and hopefully we can find some ways to use specific epigenetic perturbations in order to accelerate that. And then I wanted to come back to these guys over here on the left. So I've been focusing on things that were pro-neuronal differentiation, but when we do the combinatorial screen with NGN3 that is already pushing neuronal differentiation, we also find things that are repressing that process. And so we took a look at some of these different repressive factors, and some of these are described separately in this plot here. But what we observed was that some of these factors were actually relatively highly expressed in pluripotent stem cells. So this is looking at expression data in embryonic stem cells. And so what we hypothesized was that if these are things that are expressed in pluripotent stem cells, and when we push them higher, they repress neuronal differentiation, then maybe if we can repress these factors, that would accelerate or enhance neuronal differentiation. And so then what we did was, rather than using the DCAS9-based activator, we made DCAS9-based repressors, showed that we could deliver that with a targeted guide RNA in each of these, to the promoter of each of these genes in pluripotent cells, and repress the expression of these repressors, and then said, okay, if we target a repressor to the antagonizers of differentiation while simultaneously directing an orthogonal activator to the proneuronal genes. So now we have repressors going to some genes and activators going to other genes. Can we drive enhanced neuronal differentiation? And it appears that that may be the case. In this case, when we activate neurogenin-3 while simultaneously knocking down ZFP36L1, we get increased levels of activation of our reporter. So still more work to do to characterize this, but one obvious question of driving differentiation is, do you also need to block other pathways? And with these types of orthogonal epigenome editing systems, we may be able to do that. In addition to looking at the reporter, we also looked at gene expression for the different neuronal markers. And then the thing I want to mention in the last minute is, as we continue to develop these epigenome editing tools to guide cell fate decisions and interrogate the genome, we're also really interested in exploring the diversity of CRISPR biology. This is a probably already out of date representation of the diversity of CRISPR systems that have been found in bacteria throughout the world. And you may have seen something like this before, but you may be interested to know that while all of the focus is on Cas9 or its relative Cas12, that actually those systems, what are called the type 2 CRISPR systems, make up a very small part of all of CRISPR biology. There's actually many other types of systems, but you may notice from the schematics of these loci that some of these systems are a lot more complicated than what you find in the Cas9 system. We have a single Cas9 protein that does all of the work. But we've been interested in doing the hard work of looking at some of these more complicated systems to see, could they also be repurposed for activity for genome engineering applications in human cells. And as a starting point, we looked at the type 1 CRISPR systems. And so what's interesting about the type 1 CRISPR systems is they make up about more than half of all of CRISPR systems that have been found in nature, whereas these type 2 CRISPR systems like Cas9 actually make up less than 10% of CRISPR biology in the world. However, these type 1 systems are a lot more complicated. So there's multiple subunits, they have interesting stoichiometry, and so there's more delivery challenges and more engineering challenges, but they're certainly surmountable, and it opens up a whole new category of CRISPR systems, as well as other things like different specificities, different PAM targeting sequences, different ways to attach effectors onto these CRISPR systems. And so I'm not going to spend much time going into the details because this work was just recently published, but we've been able to take these type 1 CRISPR systems and similarly attach epigenome modifiers to them in order to get targeted activation, repression, and also cutting in the genome. And so we're excited about this as incentive for genome engineers to go and explore the rest of CRISPR biology outside of just Cas9 for new genome engineering tools. These tools are also very specific. When we turn on that target gene in the genome, we can do so with basically exquisite specificity, where it's the only gene that's significantly upregulated. So just to summarize, I wanted to emphasize kind of the specificity and robustness of these CRISPR tools for epigenome editing, not just gene editing where they're most famous, and the ability to use them for going in and interrogating the gene regulation, but also for making kind of deterministic cell fate changes with these tools. We also have used them for these types of high throughput screens of factors that are involved in cell decision making, and we're interested in expanding this CRISPR toolbox to go into other types of systems for genome engineering. So of course I want to thank the Frontiers group for their support of the work. It's really allowed us to move into a lot of these other more difficult areas, like expanding the CRISPR toolbox and unbiased screens of the genome for cell fate specifiers. And hopefully I acknowledge these are the students and postdocs along the way that did the work. And thank you for your time.
We have time for maybe one question, if there's one. Jeff?
I'm wondering how you're thinking about the known dynamics of differentiation and the fact that, for example, the same transcriptional regulator can be expressed by many mutually inconsistent cell types, neuron types, that are, and expression and control is state dependent. How do you think about getting there instead of having everybody come in all at once?
Yeah, we're really interested. I mean, you know, the ideal experiment would be able to test in a pooled screen all combinations of all factors at all different levels at all different times. But you know, there's only so much that we can pull off right now with current technology in a single screen. We are thinking about that, and would like to take some of these orthogonal systems and put them under temporal control or inducible control with things like DOX-inducible or other chemically inducible systems. So we could look not just at the combinations of transcription factors like I showed there, but control a certain set of transcription factors at time 0, bring in other ones at other time points. So we'd like to look at that. It is technically challenging, and so we have some work to do, but that would be the next place that we would want to go. It's a good question. Thank you.
Okay, thank you so much.